Good morning or good afternoon, everyone. Welcome to the continuation of the series of seminars by each of the NAI teams. This is Origins of Life Week here at NAI Central Plaza. And we're going to hear today from John Peters for the Montana State team. John got his BS in microbiology at the University of Oklahoma in Norman and a PhD in biochemistry from Virginia Tech, and then was a postdoc at Caltech before moving on to MSU. John is also the director of the Thermal Biology Institute at MSU, in addition to being the PI for the NAI team. And the Thermal Biology Institute investigates the geothermal environments of Yellowstone National Park, which the NAIEC will have the privilege of seeing and being shown by John in September at our next in-person EC meeting after the ASU workshop. And we're all looking forward to that. And without any further ado, I will turn the floor over to John.
John, I think your microphone's muted. Is that true?
Out there.
Okay, now we can hear you.
Okay, thanks, Carl. Can they hear me now, Marco?
Yes.
Okay, you said something about the delay. If I'm not observing the delay, can you reel me in, Marco, please?
Sure.
Okay, thanks. Okay, so I'm going to talk about our project that's really centered on structure and reactivity at iron-sulfur motifs. And I've got a number of my PIs here with me, so and I'm going to try to field the whole center's thrusts by myself, but if I see them waving their arms or rolling their eyes or something like that, I may call on some of my PIs to clarify some things that I'll be discussing. Okay, so our team is a number of investigators here at Montana State University, Martin Schoonen and John Parisi at Stony Brook University, and Dan Strong at Temple University. And it's a interdisciplinary team of different flavors of chemists mainly, and so we have biochemists or biochemists, and all the way to computational and physical chemists, or we're spanning that range of chemistry. So Joan Broderick and I our expertise is in metallobiochemistry, Martin Schoonen in geochemistry, and John Parisi in geochemistry as well, Dan Strong in surface science, Trevor Douglas in nanomaterial science, Tim Minton in molecular beam science, Mark Young, a molecular biologist and virologist, Robert Sloggy, a spectroscopist and computational chemist, and a philosopher, Prashanta Bandyopadhyay. Pretty good job with that one, I think. Okay, so it's a fairly focused center where we're probing the relationships between structure and reactivity of iron-sulfur motifs, and the role of iron-sulfur motifs in prebiotic chemistry and the origin of life. So there's been a lot of ideas about the origin of life, and those have evolved with time, so to speak. And I think without going through this slide, our project is really can be tied to metabolism first ideas for the origin of life, but are really focused more on prebiotic chemistry, so relevant to RNA world, lipid world, making protobiomolecules of any kind. But I think, Wachtehauser's ideas on the iron-sulfur world and Mike Russell's ideas, I think that our project is in line mostly with those ideas. So what we're interested in the reactions of raw materials important for the production of protobiomolecules or prebiotic chemistry. And I've listed some of those molecules, those small molecules here, and in the a hypothetical origin of life broken into 4 stages here, a Hadean stage in which we have interconversion of small molecules to increase the pool of raw materials for condensation reactions in the second stage, which is followed by polymerization reactions, and then organization here. And so I haven't drawn any line here of where life emerges, but we all know that, at this stage when we have cells and organization, we have life. So the interconversion between small molecules, these small molecules that I've shown above, maintaining the pool of raw materials for condensation reactions, the second stage that result in the formation of protobiomolecules, is critical for the origin of life. So again, we're talking about Hadean chemistry of this first stage. So when Dave Desmarais gave his talk, he presented this slide from Andrew Porrillo about the integration of origin of life research across the Institute, which includes the studies at Goddard, origin and evolution of organics, studies at Rensselaer, the catalytic potential of clays, and then to Carnegie, the geochemical steps leading to the emergence of catalysis. Then our project, origin and evolution of iron sulfur protein enzymes, the Ames group's research on the origin and evolution of enzymes, and then the Georgia Tech group looking at the ribosome and RNA and protein translation. And I think, now that I have this in my hands, I can say that, I think our project really, in addition to focusing on this area, really spans from these studies on the catalytic potential of things through the origin of, and evolution of enzymes. Okay, so in biology, Hadean chemistry or the like is catalyzed by iron-sulfur enzymes and some complex iron-sulfur enzymes. And iron-sulfur proteins and enzymes are ubiquitous in life. Almost all organisms have these smaller, simpler versions, 2 iron 2 sulfur clusters, 4 iron 4 sulfur clusters, 3 iron 4 sulfur clusters. In many cases, those clusters involved in electron transfer reactions, are involved in photosynthesis, in respiration, involved in many other processes. But we also have these more complex versions, like the metal clusters associated with nitrogenase, the P cluster, iron, molybdenum, cofec, the metal cluster associated with the iron-iron hydrogenase, or the H cluster, and these clusters associated with the carbon monoxide dehydrogenase acetyl-CoA synthase complex, which has nickel iron-sulfur clusters in a couple different forms. So those reactions catalyzed by these complex iron-sulfur cluster-containing enzymes, again, these are reactions, Hadean chemistry, or reactions that would have been important in the early Earth, or at least reactions that are very similar to the reactions that would have been important in the early Earth, like the hydrogenase reaction, the reversible oxidation of hydrogen, the nitrogenase reaction, the reduction of nitrogen to ammonia, the interconversion between CO and CO2, or the condensation of CO and methyl groups to form activated acetate. So iron sulfur minerals and other derivatives have interesting structural relationships with enzymes and have also been shown to catalyze these type of reactions. And this is a slide from I've unintentionally created a strange animation here, but this is a slide from Mike Russell's work, and Mike Russell and Bill Martin. Just looking at the similarities between the clusters, the active site clusters of CO dehydrogenase and acetyl-CoA synthase, and the structure of a nickel-containing greigite mineral lattice. And so you have nickel, these themes, the nickel and iron-sulfur clusters in association. And, we would assert that the relationship between iron-sulfur mineral and iron-sulfur enzyme structure, function, and reactivities just too strong to be coincidental. And especially with the, what we know, the abundance of iron-sulfur minerals at the origin. Okay. That's an example of structural relationships. Here's a couple of examples of reactivity in iron-sulfur minerals or the like. This is an example from Günter Wachtehauser, looking at the formation of activated acetate under primordial conditions with small molecule precursors and a nickel iron sulfur amalgam as a catalyst, forming an activated acetate of this proposed mechanism. This is an example from our co-PI Martin Schoonen, where he's been able to show that he can get nitrogen reduction under hydrothermal vent conditions, or really, if you compare to the conditions where we produce nitrogenous fertilizers, milder conditions in temperature and pressure. And so on this slide, just showing that the rate of nitrogen reduction to ammonia with hydrogen sulfide is increased in the presence of pyrite. And Martin and a number of other investigators have been able to show that defects in mineral structure actually enhance the reactivity or enhance the catalytic activity of minerals. And I'll get back to this. So those three examples really implicate iron sulfur, or the role of iron sulfur minerals in these two stages of the origin of life in this four-stage model that I've already shown you. And when you think about Marco, am I doing okay with the delay?
Yeah, it's fine.
Okay. So, if you think about iron sulfur minerals serving as catalysts for these varying reactions, then you have to think of a variety of iron sulfur minerals. You showed, the Greigite example. That's one modification, nickel association with iron sulfur minerals. But, to catalyze nitrogen reduction or reversible hydrogen oxidation, you have to think of a variety of different minerals. And I think that, hydrothermal vent systems or the effluence of iron sulfur-rich thermal springs are really inspiration for this idea, because you have gradients of temperature and pressure and different gas solubility, mineral solubility, really combinatorial chemistry with respect to the types of iron sulfur minerals that you could generate in these systems. Okay, so our project overall is really asking this question: can we think about the active sites of iron sulfur enzymes as highly evolved derivatives of iron sulfur minerals? And from that, we have, and I'm showing here the active sites again at the H cluster of hydrogenase and the iron molybdenum cofactor of nitrogenase, a couple of our favorite model systems with the structure of minerals and a mineral lattice. And from that overarching question, we have a number of different questions that we can attach experiments to, including do mineral modifications enhance reactivity? Can minerals be modified in a manner inspired by the biochemistry to enhance reactivity? What is the influence of organic nesting or the protein environment on structure and catalysis? What are the structural determinants of catalysis and how they're derived? How are modifications chemically conferred? What is the evolutionary history and origin of iron sulfur enzymes? And so this really, these list of questions that I just rattled off for you really represents sort of a continuum that's going across scales. And that's really, in a way, how our project is set up in three thrust areas. The first thrust area focusing on iron sulfur mineral and modified iron sulfur mineral structure and reactivity. That's our mineral thrust. A bridging thrust, a mimetic iron sulfur cluster synthesis and organic nesting thrust, probing the transition between abiotic and biotic, or the relationship between abiotic and biotic iron sulfur catalysis, and that's our mimetic thrust. And then iron sulfur enzyme structure, reactivity, and biosynthesis are enzyme thrust. So for our mineral thrust, really the hypothesis, the overarching hypothesis, metal centers in enzymes are more likely related to mineral defects than ideal mineral structural motifs, and really sort of probing the effects of defined modifications or defects. I hate to, I work on these beautiful enzymes that have these beautiful iron sulfur clusters. I have a reluctance to connect them to defects, so they're modifications. Examining the relationship between defects, the structure of defects, and the effects on reactivity. And so within that, we have three different levels of which we're looking at minerals. One is to synthesize these mineral surfaces, modify those mineral surfaces, or if we must, introduce, define defects in that, characterize the nature of the defects, use a combination of advanced experimental and theoretical techniques to develop insight into the reactions, look at the reactivity directly and the intermediates in those reactions, and then aim looking at defining the energy barriers of processes connecting the reaction intermediates together, really defining the reaction mechanism for minerals. And a lot of this mineral thrust has focused on reactivity dinitrogen reduction. It's sort of the tour de force. This one is the most challenging reactivity. Okay, so from this slide, I've already shown you what the mineral thrust is focusing on is these two questions here: do mineral modifications enhance reactivity, and can minerals be modified, inspired by the biochemistry, to enhance reactivity, or really addressing this ligand-accelerated catalysis? Okay, so the first, generating these mineral surfaces or defected mineral surfaces and characterizing those. That's really a thrust that Dan Strongin's spearheading, and so he introduces these defects, and you can see here hydrogen, barring hydrogen on this mineral surface, hydrogen sulfide being a product, and then characterizing those defect sites by synchrotron-based photoemission or even photoelectron spectroscopy in the lab. The next part of the mineral thrust is looking at reactivity directly, spearheaded by Martin Schoonen, who's looking at nitrogen reduction reactivity, but also nitrate and nitrite as sources for abiotic ammonia, the reduction of those molecules as well. He's, in this first part of the grant, really developed a system to, in a very sensitive way, detect nitrogen reduction, or a sensitive method for analyzing ammonia using this, making this fluorescent derivative, and has a flow system where it can analyze the samples. He and Dan Strongin are teaming up to look at intermediates in those reactions using IR spectroscopy, and a significant challenge with these is or a barrier that had to be overcome was just with the sample apparatus having hydrogen sulfide around. It's very corrosive. There were some problems with the sample apparatus now, and now this sealing, the support by gold plating has actually allowed them to overcome those challenges and are generating results. So the third part of this, defining energy barriers of the processes and connecting reaction intermediates, is a thrust that's spearheaded by Tim Minton and involves molecular beam science. And here's the instrument that Tim uses for these studies, the molecular beam studies. And Tim joked to me that if he wouldn't have become a physical chemist, he would have become a plumber. And I think those skills as a plumber really come in handy in terms of maintaining this instrumentation. Now, see, I did get my co-PIs to roll their eyes at me at that one. Okay, so what Tim's looking at, models of prebiotic chemical transformations, mechanisms for hydrogen and nitrogen reactions at iron-sulfur mineral surfaces. So really bringing together the results from Dan and Martin's work and focusing on energy. So a molecular level description of the mechanism of nitrogen reduction will guide our understanding of what kinds of surfaces are likely to be reactive. And so these studies will provide an input for theory and experiments by other groups, explanation of the observed phenomenon in complex experimental systems, and insight in the structural motifs that have been seen in nature. And so really what we're talking about is understanding reaction at the molecular level with these studies. And what Tim has is, really it's basically a closed system where you can modulate a lot of different variables, and some of the variables are shown here, and look at what comes out at the end with mass spec, and then from that infer what's going on at the surface. You can look at the inelastic scattering, which will be indicative of energy related to bond breakage and bond formation. Okay, and here's an example of experiment here, bombarding or using a molecular beam deuterium atoms on a pyrite surface and looking at the formation of D2S, which been looking at D2S, and that's shown here, and this Maxwell-Boltzmann distribution indicates that the formation is in thermal equilibrium. And then here is a set of images showing what happens to the mineral surface after exposure, 5 minutes and 320 minutes here. And these are SEM images of that surface. And Robert, who's not with us today, Robert Szilagyi, one of our co-PIs, is actually at the synchrotron source analyzing what's happening at this mineral surface using XF, so what's happening to the bonding of iron and sulfur at this mineral surface. So Tim has also been able to show that the evolution of nitrogen scattering on these surfaces, these modified surfaces, is affected by the surface roughness. And so this may tie us back to what we were saying earlier about that the defect sites or defects affecting reactivity. Okay. I got through that one. That one's actually the toughest one for me. And on to the next thrust, the mimetic thrust. And this is Trevor Douglas, the leader of that thrust, involves Trevor Douglas, Mark Young, and John Parisi at Stony Brook. And so this is iron-sulfur cluster synthesis or organic nesting. So this is really looking at the effect in a controlled way of a protein environment on the physical and catalytic properties of metal clusters. And the systems they use are protein cages as templates for the synthesis of clusters, and they can do this in a controlled way. This is one example of the ferritin molecule from Pyrococcus furiosus. It's a thermostable template that they can use for synthesis, which gives greater flexibility and actually do these syntheses under hydrothermal vent conditions. Okay, and so that thrust, the mimetic thrust, spans these questions in our continuum of questions looking at the relationship between iron-sulfur enzymes and iron-sulfur minerals. And so the synthesis, and I'm showing this, the synthesis of or the production of iron oxide clusters here in this mineral cage. So in this EM picture, we have the cage, and you can see this is a stained image where you can see the protein here, and then these are unstained images where we're looking at the density of the metal clusters. Here, iron oxides, and in here, iron sulfides. And iron oxides can be produced by the procedure shown here, and iron sulfide or iron oxides can then be converted to iron sulfides. These clusters can be characterized in a number of ways, and this is an example of characterization by small angle X-ray scattering and pair distribution function, which allows us to look at these clusters. They're really they don't have a well-defined mineral lattice, and so the bonding isn't uniform. And so this approach, which gives us similar information to the XF somewhat, and that we can get the bonding of iron and sulfur in these somewhat heterogeneous mineral clusters. And also control and monitor the cluster size. And again, this is really an organic template. We're growing mineral clusters and then looking at the effects of cluster size and the protein environment, what the protein environment imposes on the clusters in terms of its physical properties and reactivity. This is one example of an effect of the protein environment on the properties of these mineral clusters, and this is looking at magnetism, and this is showing here a cluster with the protein environment here showing that in the presence of the protein environment in this particle all the spins align, but if we remove the protein environment here, the spins at the surface randomize. And then can introduce that protein environment and restore that the alignment of those spins. So the protein environment impacting the physical property of those clusters. Another example here is looking at the effect of size on the physical properties of those clusters, and this is again an example of magnetism with particles 5 nanometers, 8 nanometers, and 20 nanometers, applying a magnetic field to those particles and looking at the change in moment here. And you can see in the 5 nanometer particle and the 8 nanometer particle, it returns to the same moment here, but the larger particle actually has some memory, so there's a difference in the physical property of that larger particle. So size affects the physical properties of the particle or the cluster in this case. Another example, the influence of the protein environment on catalytic activity, and this is an example of EPIC 203 in ferritin, and looking at that cluster in terms of its reactivity as a photocatalyst, and actually being able to measure this as a photocatalyst. And so that's an example of reactivity, a reactivity that we can measure, and that's an objective in the thrust. And now, go back to that slide. We have a facile means to convert these iron oxide clusters to iron sulfide clusters, and the parallel analysis is now ongoing for the iron sulfur clusters, and I have some data on that, and that will soon be ready for prime time. Okay, so that's the second thrust, and I'm actually doing pretty good on time. I'm amazed, but I think I'm going like a mile a minute here. And I can go into our iron sulfur enzyme thrust. And so this is a thrust that Joan Broderick and I are spearheading, and what we're doing is probing the structural determinants for catalysis. We're probing complex cluster biosynthesis and the evolutionary origin and history of iron sulfur proteins and enzymes. A couple of our favorite systems, the iron-iron hydrogenase. So this is an enzyme that has a number of different metal clusters. One of these complex iron sulfur clusters is a 4-iron 4-sulfur cluster that's bridged to this really organometallic 2-iron unit that has carbon monoxide and cyanides as ligands to iron. These hydrogenase systems are the only known systems that have this variation of modification to an iron sulfur cluster, these diatomic ligands. And then nitrogenase, and this is the protein that harbors the active site for nitrogen reduction to ammonia, the iron molybdenum cofactor. And so this shows the protein environment here. This is a monomeric protein. Has these 5 different metal clusters. It has these accessory clusters that were presumably involved in shuttling electrons to or from that active site in reversible hydrogen oxidation. And nitrogenase, which is a heterotetramer, 2 types of subunits, 2 types of metal clusters, one the P clusters, which are again intermediates in electron transfer, to the business site, this iron molybdenum cofactor, which has it's a complex iron sulfur cluster, but it's heterometallic. It has molybdenum in addition to iron and sulfur, and again some organic groups that are present to tune the reactivity of that cluster. Okay, so now we're in this thrust moving through this continuum. What are the structural determinants of catalysis and how they're derived? Really, what are the structure and reactivity of these enzymes? How are the modifications conferred? That's the biosynthesis. And then what's the evolutionary history of the origin of iron sulfur clusters? Okay, so in this slide I'm just showing what's kind of known about the biosynthesis of the iron molybdenum cofactor in nitrogenase. And interestingly, that making a complex iron sulfur cluster, these unique modifications to the variations on this complex or this iron sulfur cluster theme requires an enzymatic pathway, requires a lot of biochemical machinery, requires scaffolds and carriers and enzymes and a bunch of things that are thus far unknown. This slide really isn't from our work here. This represents about 30 years of work from a number of different investigators to really at least get this level of insight on iron molybdenum cofactor biosynthesis. And so what this slide is showing, just to simplify it, is you have a scaffold, and a scaffold that's structurally similar to the molybdenum iron protein, where a bunch of different enzymes and carriers act to build an iron molybdenum cofactor. And after that iron molybdenum cofactor is built, that's inserted into an organic nest or an apoprotein to give it its reactivity. So this is really interesting in terms of the prebiotic chemistry and the way we're thinking about things, because you have the apparatus for the cluster is really separate to building the nest, and in the final step, to make the cluster reactive, you insert the cluster in the nest to make an active enzyme. Okay, so there's some really interesting ties between the biosynthesis of the cluster I just showed you, the iron molybdenum cofactor, and our hydrogenase system. And that link is in iron sulfur-based radical chemistry. So there's two enzymes in the biosynthesis of this cluster, the H cluster, and one enzyme involved in the synthesis of the iron molybdenum cofactor that are radical SAM enzymes. They're iron sulfur-based radical generating enzymes. And so this is the active site of that enzyme, of this whole class of radical SAM enzymes, which hide E, hide G, and NIF-B are members, where you have an iron sulfur cluster and S-adenosylmethionine, and that iron sulfur cluster cleaves S-adenosylmethionine to form methionine and a radical. And that radical is coupled to a lot of different chemistry in nature, generating radicals of amino acids. It can be intermediates. A radical intermediate of an amino acid is involved in amino acid isomerization reactions. It's the reaction for the radical generation is important for sulfur insertion, like the sulfur insertion to biotin, it's shown here in this biotin synthase. Sulfur insertion for generating lipoic acid. The known radical SAM chemistry really sparked us to kind of generate a hypothesis how that type of chemistry could be involved in hydrogenase H cluster biosynthesis. And as I mentioned, that's a really interesting variation on the iron sulfur theme that has very unique ligands. In that, and we can see this up here, we have a dithiolate ligand, only place in nature that's seen. It's a non-protein dithiolate. This isn't cysteines, this is a thiolate ligand that's not associated with the protein. And these carbon monoxides and cyanide ligands, again, a unique feature to hydrogenase. So we started to ask the question, how could those ligands be generated by radical SAM chemistry? And we thought about that in the context of known radical SAM chemistry and came up with a hypothesis. A hypothesis that if we started with a simple iron-sulfur cluster precursor, we could introduce a dithiolate ligand from a central metabolite, amino acid, or glycolytic intermediate. We don't know the substrate for that yet. By chemistry that's very similar to known sulfur insertion chemistry, chemistry that we know that these radical SAM enzymes will catalyze. And we could generate an intermediate that actually shifts the reactivity in this small iron-sulfur cluster to the iron atoms. And we can think about reactivity occurring at the iron atoms and the generation of these diatomic ligands, these unique diatomic ligands. And then again, we go back to this known reactivity for the radical SAM enzymes, and we've proposed that if we formed an amino acid radical through this chemistry, that at that iron dithiolate or iron thiolate site, that we could get decomposition of an amino acid radical to form carbon monoxide and cyanide. And if you think about that, if you think about glycine, if you take glycine, you bind it at an iron thiolate site, a glycine radical, if you shuffle around the electrons, you can generate carbon monoxide, cyanide, and water. Very similar to the ligand arrangement we see in the iron-iron hydrogenate. So this is a hypothesis, and it's very intriguing in terms of astrobiology. And Robert Szilagyi generated a potential energy surface to map this reaction. And if we have an iron-1 thiolate, and this is an in silico experiment here, we can generate a potential energy surface or a reaction coordinate that describes a favorable reaction of the decomposition of glycine to carbon monoxide, cyanide, and water at this iron thiolate site. So we have a favorable reaction, we have an activation barrier, but the activation barrier in this reaction is generating that amino acid radical. And we know that we can overcome those barriers with radical SAM enzymes. So this is, if you think about this reaction, you have the decomposition, going in this direction, the decomposition of an amino acid to form carbon monoxide, cyanide, and water. If we think about the reaction in the reverse, which we can get a favorable reaction in reverse if we monkey with the oxidation of the metal side of the iron thiolate, then we're talking about a condensation reaction where we have CO and cyanide and water to form an amino acid. And so really this represents another potential link involving iron-sulfur motifs, linking Hadean chemistry and abiotic chemistry, or interconversion between small molecules and condensation reactions. Okay. Again, in these studies, and I'm just going to talk about a few examples of what we're doing in structure function of nitrogenase and hydrogenase. And, an interesting, and I've already drawn your attention to this, is an interesting observation that for nitrogenase, you have the synthesis of the iron molybdenum cofactor occurs on a scaffold. And we've been examining this process, or the synthesis, just showing you that hypothesis for iron-iron hydrogenase cluster. And a lot of the ideas that we generated in terms of developing an experimental line to probe H-cluster biosynthesis was really based on some of the observations in nitrogenase biosynthesis. And one of the questions that we've been asking is that theme carry over to hydrogenase? Do you make a cluster independent of the protein on a scaffold and then insert the cluster into the organic nest to define its reactivity? And we have some new results where we're looking at the structure of that apoenzyme, or the hydrogenase, before it's been acted on by those biosynthetic enzymes. And what we've been able to show is that apoenzyme is devoid of not the whole cluster, but just that 2-iron unit. And from that, we can get the insight that all this machinery is directed at just that 2-iron unit, just that organometallic-y thing with CO and cyanide and dithiolate linkage. And that's kind of what we had proposed in this hypothesis, because these iron-sulfur clusters, these 4-iron 4-sulfur clusters, which is the other component of that cluster, are typically made by host, housekeeping cell machinery. But what we've been also able to show is, in showing the apoprotein that's poised to accept this 2-iron unit, that there are really just amazing similarities between this hydrogenase and the nitrogenase. In the apoproteins of both of those systems, you have a channel leading to the active site, ready to be the recipient of this cluster. It's a positively charged channel. Here we have the hydrogenase and the positively charged channel, and the nitrogenase, its positively charged channel. And both of these metal clusters, without the influence of their protein environment, are negatively charged clusters. So amazing parallels between the two systems, and a general theme in terms of this final step, the organic nesting of these catalytic clusters. Okay. Another, our study, or this scheme for nitrogenase FeMoco biosynthesis is really kind of inspiration for our studies aimed at looking at the evolutionary origins of nitrogen fixation. As I mentioned, this NIF-EN, or this scaffold protein, is structurally similar to NIF-DK, or the hydrogenase. And so this was generated, this scaffold, in defining the entire process, or getting an active nitrogenase. This was generated from a gene duplication event, where DK was duplicated as a unit to form EN. And so we've been using that gene duplication event, as well as gene fusions events, or the traits of 7 different genetic loci, to trace the evolutionary origin of nitrogen fixation in extant organisms. And this work was just submitted for publication, and I don't have time to go through and describe all the facets of the work, but basically the bottom line is that we can say from the occurrence that nitrogen fixation was not likely to be present in the last universal common ancestor, and that nitrogen fixation, or at least molybdenum nitrogenase catalyzed biological nitrogen fixation, originated in methanogenic archaea. And I think, this line has been of interest to the Arizona group and the work of Bob Blankenship and Jason Raymond, who's part of their new team. But also, where do we find nitrogen fixation, or where do we find hydrogen metabolism? Where were these processes likely to emerge on Earth? Are topics that really bridge the interests of our team, the Arizona team, Ames, Penn State, with Greg Ferry, the Carnegie team, of which John Barros is a member. So there are a lot of good connections to make in these areas. Okay, so for our experimental to kind of come to a conclusion with our experimental line in terms of our project, really, Robert Szilagyi he's a computational chemist, a theoretical chemist, a physical chemist that is really using theoretical chemistry and X-ray absorption spectroscopy to bridge all our 3 thrusts together. So Robert is able to use one approach, or these 2 approaches, theoretical chemistry and X-ray absorption spectroscopy, to analyze all materials across the continuum. So he can use one approach to look at all these different things that we're looking at in the mineral thrust, the mimetic thrust, and the iron-sulfur enzyme thrust. We have a very strong philosophy component and have been developing a strong philosophy component. And in these origin of life studies and origin of catalysis, there's a number of interesting questions that can be posed philosophically. The traditional questions, the analysis of RNA versus metabolism debate, the analysis of those questions, whether we're asking the right questions, whether we can use traditional philosophical approaches to analyze that question. And that really brings in, can you use traditional philosophical approach to take a look at emerging sciences and reviews and theories about the definitions of life, really creating a framework to pursue philosophical questions related to the nature of living things. And this group, from shortly after our project started, we had a weekly working group for the philosophy component of this, and this group consists of students in philosophy and students in our science programs, graduate students and undergraduate students, students and faculty. And this has really been a powerful tool for all of us in the center thinking about how we think about our problems. We've traditionally at MSU had a strong outreach program in astrobiology relevant areas with our Thermobiology Institute, and we're continuing that tradition in our outreach program, really now kind of catering that to this new thrust of prebiotic chemistry. And we have a lot of success using this train the trainer molecule and model. I'm stuck on molecules. It's a train the trainer model where we have a number of classes aimed at science teachers, and mainly science teachers at the middle school level. And so this is part of their curriculum and professional advancement. They're masters in science education courses. Most of them are classes that are held here at MSU, but we're developing distance learning components for these classes. We have a form of this class that's really more traditionally the Thermobiology Institute's thrust that's on more microbiology, and more the ABRC's thrust, which is on chemistry, so for chemistry teachers and for more biology teachers. We have developed a lot of online learning tools. We had this year a couple of very successful community lectures and a lot of programs there. And a lot of those programs, they can be, you can take a look at our website, and we'll be highlighting what's going on with the outreach program as well as the scientific program as things progress on our website. So I encourage you to take a look at what's going on at the website. So I'll just close here, and I can't believe it's like almost exactly 50 minutes. Close here with a slide that is really just some cover art that we've made for a review that's just come out in Dalton Transactions, which is a chemical journal, where we're kind of showing this continuum from minerals to iron-sulfur catalysis. Okay, I'll close here. I made it.
John, thank you very much. We are going to open up for questions in just a second, so if you have a question, either raise your hand in Adobe Connect, or if no one raises their hand, you can just jump in. I just wanted to make an observation, and that is that one other connection between your work and work at the other teams that struck me, thinking back to the Wisconsin seminar and Nita Sahai's description of that very nice work looking at the very detailed interactions between mineral surfaces and small molecules. Seems that the techniques that she's applying probably are applicable to the work that you're doing as well. So just struck me as one additional connection. And I suspect we're
Well,
probably...
that was one thing that I forgot to mention, and I meant to mention that, and that really also is tied to radical chemistry and some of the things. So I think that, thanks very much for making that point.
Okay, John's talk is open for questions, please, including questions from right there at Montana State. I could imagine with all those folks in the room, there might be a question or two for you, John.
I think we have one from Lauren Williams.
Okay, Lauren, please go ahead.
That caught me by surprise because I didn't really have a question formulated.
Oh, okay. I saw your hand raised.
That might have been. Sorry.
It sounds like looks like George Cody has a question.
Okay.
John, that was a wonderful talk. Thank you very much. This radical SAM chemistry is really something that's intriguing and not something that I know a lot about, so I'd like to talk to you about in the future. But I just had two questions. The enzymes associated with this radical SAM chemistry, do you find that these tend to be a conservative group of enzymes? And the other is, are there any metallocenters associated with these enzymes? So is there transition metal chemistry associated with the radical SAM chemistry for the assembly of the H center, for example, in hydrogenase? That's my question.
Joan, do you want to? Okay. So they have a 4-iron 4-sulfur cluster is the inorganic part of those enzymes. They all have a 4-iron 4-sulfur cluster, and it's a site-differentiated 4-iron 4-sulfur cluster where you have 3 of the irons have cysteine ligands, and the other ligation is provided by the S-adenosylmethionine. And then, as a group, evolutionarily, they're not all that conserved. There's enough conservation to actually identify them and annotate them as a class. If you look at the enzymes and the way they're annotated in terms of their function, most of them, no, I won't say most of them, many of them are incorrect, and so they're hard to put into their different functional classes. But we're looking at that, and we're trying to look at the evolutionary trajectory of those radical SAM enzymes, and like in the context of lip-A or bio-B or some of these other enzymes involved in isomerization of amino acids, and our enzymes involved in cluster biosynthesis, we're trying to look at the evolutionary relationships there, which I think will be really pretty interesting.
We have a question from Jen McAlady.
Hi, John. Thanks for your nice overview. I have a question you may or may not want to speculate about, but I was just reminded this weekend about Paul Davies' ideas about weird life. And it strikes me that if iron-sulfur chemistry was a feature of the Hadean world, you might expect both normal life and weird life to have iron-sulfur catalysis. Have you thought about that at all?
No, I haven't. That's interesting. I'll think about that. I'm not sure what to say about that.
We have a question from Barry Bloomberg here at NAI Central. Barry, you can just go right ahead.
Thanks for that talk. How does this relate to the formation of magnetosomes that occur in bacteria, archaea, and other organisms? Is there some understanding of that connection?
Yeah, Trev, do you want to say something?
Yeah, so there definitely is on a mechanistic level. There's a similarity. Those are deposited. What we know about them is that there are metal transporters that pump metals across a membrane into a vesicle, and those things are grown from very, very small particles into their final state and are confined by the vesicle. And they make both an iron oxide and an iron sulfide, and sometimes a mixed oxide-sulfide phase.
How high up in the evolutionary scheme do you find the formed magnetosomes?
Ooh, John.
Yeah, I don't know the answer to that.
I don't know that.
Thanks.
We have a question from Goddard.
Yeah, hi. That was a very interesting talk, and I wanted to ask you a question which isn't directly related to the mineralogy you're doing, but more related to the use of a phrase my question will reveal my own ignorance, and so I hope to be educated. And the question is, what do you mean by evaluative methods of the philosophy of science?
Yeah, well, I'll have Prashant address that.
Well, what we do in philosophy of science, we have some standard criteria to evaluate two competing theories. For example, the scope of a theory, the fruitfulness of a theory, and the predictive powers of a theory. But when you look at a finished theory, for example, general theory of relativity, so we have a very specific way to evaluate the theory, whether the theory is better than the Newtonian theory of gravitation. But in the case of emerging science, like, when you're concerned with the metabolism first theory versus the RNA world hypothesis, so it's very difficult to apply those criteria in knowing which one is a better theory than the other. So we are hoping that by analyzing, by looking at those different hypotheses like the RNA world or the metabolism first hypothesis, we as a philosopher of science would like to know what philosophy of science can learn from also astrobiology, not also the other way around.
Well, of course, I'm very familiar with the standard scientific method for drawing predictions from a hypothesis and then testing those by going into the experimental world and making measurements that can in fact quantify those predictions and thereby iterate the theory or hypothesis. I'm less clear on how one does this in the philosophical world other than through discourse. How do you do it?
Well, the standard philosophers of science, the way scientists evaluate why general theory of relativity is better than the other. So there are like three classical tests, right? One is the bending of light case. Then we have the Mercury's perihelion precision explanation. So those are some of the classical tests, scientists use, to explain whether general theory of relativity is successful in explaining the theory. So similarly, the question is whether we can apply those criterion, better explanation, better confirmation in evaluating two competing hypotheses like the metabolism first versus the RNA world hypothesis. It's very difficult to, pinpoint their predictions and, because we are mostly doing the speculative explanations and all those kinds of things.
Thank you.
Okay.
Does
This
anyone
is
else
Lauren.
have
I
any?
have a question. I could get in.
Please go ahead.
Hey, John, I enjoyed your talk. I'm now ready to ask a question. And I think maybe this has been asked before, but so clarify it for me if you would. If these metal clusters are so ancient, and I would assume that would mean the proteins that bind them should be ancient, so what kind of sequence conservation do you anticipate? What's predicted by that and what's observed? I'm just thinking, of course, always in terms of ribosomal RNA genes, and which are ancient, and you can see that. And so how would you compare that to these proteins, to find these clusters?
Well, I think, that's an interesting question because I kind of just closed with an example saying that they're not ancient, or at least that, there's one example for nitrogenase. Our results indicate that they're not present, that they're not likely to be present in the last common ancestor and entered later. What we're talking about is you've got a number of different mineral catalysis going on. Some of that mineral catalysis is providing the raw materials, maybe the raw materials for life forms, and eventually those are taken up and made up into biological processes. Of course, I'm speculating and kind of representing our point of view. For maybe hydrogen metabolism, that happened earlier, and maybe it is present in the first life forms, and we'll have to figure out an approach that we've used that's similar to what we use for nitrogenase to ask those questions, or for acetyl-CoA synthase, or for CO dehydrogenase, etc. So I don't think, they don't necessarily, that we see relationships between mineral catalysis and iron-sulfur enzyme catalysis doesn't mean to say that those were the first enzymes necessarily. I think that probably find a subset of those, or maybe predecessors for those perhaps were, but they don't have to be. Does that make sense?
Yeah, I got that. That's very good. Thank you.
We have a question at Penn State.
Hi, John. This is Chris House. I was just curious if any iron-sulfur clusters are known from viruses.
From what I know, no. And I'm getting shit from my virus colleagues. They say no as well.
So John, that's not totally true. That people that we don't actually know. There are lots of viruses that carry these algal viruses, for instance, carry photosynthetic genes. I don't know that anybody's actually looked.
Well, so I think that the answer to the question is no. Nobody's made that connection, and nobody's found an iron-sulfur cluster containing protein in the virus. For standard like ferritoxin, like motifs and stuff, which people have looked at fairly extensively, nobody's found that either. So I think that there may be, but nobody's found it yet.
Okay, are there any other questions for John? John, I would just make the observation that in response to your last comment about the enzymes may not be ancient, by which I think you meant that they may not have been present in the very first life forms, methanogenesis, where you suggested they may have originated, may very well be an extremely ancient metabolism. So it could have been not all that long after Luca, conceivably.
Absolutely, and I think that, that's something that our studies haven't, we haven't put any sort of clock on these, and that's something that we can do, and we're trying to learn to do.
Any other questions for John? Okay, well, let's thank him again. As I think you all know, we'll continue this series on Wednesday of this week with Lauren Williams also talking about the origin of the earliest proteins, particularly how the ribosome came to be and how RNA learned to make them. So come back Wednesday. See you soon. Bye-bye.